This presentation will be an analysis of waste minimization within the pharmaceutical industry. Pharmaceutical waste contains high biological oxygen demand, chemical oxygen demand, volatile organic compounds, and pharmaceutically active compounds, including toxic substances, hormones, and antibodies, increasingly threatening the health and existence of humans and the ecosystem. So although good manufacturing practice regulations are encouraged within the industry, the waste emissions are not considered important enough to quantify. Thus, manufacturers rarely report their emission production from pharmaceutical processes, and the transport and storage is not reported at all. Additionally, pharmaceutical companies are not environmentally incentivized to uptake waste-free options due to the financial benefits provided by creating solvent waste. Thus, the implementation of waste minimization within the industry can reduce the production of waste streams and can significantly reduce the toxicity within our aquatic environment, health, and lower economic costs. Procedures within a pharmaceutical production line will always use the same general weighing and milling steps, which are dispensing and weighing each ingredient, milling to size, mixing of the powder blends, granulation, drying, compaction, and often coating and packaging. Where they vary is how the materials are mixed and ultimately pressed into shape, which is based on the type of drug being produced and the properties of their raw materials, resulting in either direct compression, wet granulation, or dry granulation production lines. In each mixture, there is an active pharmaceutical ingredient, the binder materials, and the packaging materials. The valuable starting raw active pharmaceutical ingredients can be natural leaves, flowers, fungi, or seeds, synthetic compounds, or biologics, and are usually toxic. The solvents, particularly organic solvents, which are used for reactions and purifications, compile the largest proportion of hazardous gas, liquid, and solid waste within the process. The pharmaceutical industry is a $1.3 trillion industry, which is reflected in its immense waste and pollution generation. This has resulted in the world's highest waste generation rate per unit of product, generating anywhere from 25 to over 100 kilograms of waste per kilogram of unit. This was apparent in 2013, where the US pharmaceutical industry reported over 90 million kilograms of waste to the toxic release inventory. What is especially concerning about this number is that commonly used chemicals such as acetone and ethanol are not included in this figure. So it's safe to assume that the reported figure is a vast underestimate of the true amount of waste produced. Secondly, most effluents within the pharmaceutical industry contain organic solvents, namely cyclic compounds, alcohol, and ethers, and more, as they are extremely useful in the production process. Proper treatment and disposal of these compounds is necessary as they can pose risk to human health, antimicrobial resistance, and the environment. Finally, manufacturing waste only constitutes 2% of pharmaceutical waste. 88% of waste enters the environment after patient use and excrement into our waterways. Three areas of waste minimization opportunities within the pharmaceutical industries firstly involve effective management of inventory storage and categorization, which can limit excess, expired, or redundant materials. Secondly, modification of production and operational procedures. This can be actioned through reducing raw material wastage and spillage, or improving cleaning and maintenance of equipment to increase optimization and longevity and lower maintenance costs. Finally, an implementation of modified equipment along the process line that will assist with on-site waste treatment or reduction and material recovery, such as vapor and water recovery systems, will enable opportunities to treat and reuse vital materials in other areas of the production and lower waste material costs.
To achieve waste minimization in the pharmaceutical industry, source reduction must occur by way of changes in products, raw materials, processes, technologies, or procedural and organizational functions. This can impact products, giving them undesirable effects such as taste, color, and dosage requirement, and thus impacting the customer buying the product. However, it is also proven to help reduce the cost of manufacturing drugs. Solvent use consistently accounts for between 80 and 90% of mass in a typical batch process for pharmaceutical production, as shown on the right. This is concerning as most, if not all, common solvents display toxicity or harm to the environment. Solventless reactions are viable, but cost is the main issue, and non-traditional solvents are often very specific, Greener solvents are often not considered as the most common and easiest solvents are usually used and any byproducts or hazards are just dealt with. If we look at data such as the solvent usage list from the UK pharmaceutical company GSK, it suggests there is a trend towards decreasing common toxic solvents like THF, toluene and DCM, but this does not mean that they are not widely used. This ties into the product/process yield, which determines the product to waste ratio. Process efficiency can be improved by increasing automation to reduce human error, which allows for stricter control mechanisms to be placed on machine operating. Installing internal recycling systems for cooling waters and solvents, and modifying tank and vessel dimensions can also greatly improve operating efficiency. Another major issue arises in finding suitable replacements for dipolar aprotic solvents, that is, solvents with strong permanent dipoles and low reactivity, as they are hard to find. Green chemistry is being rapidly adopted by process chemists and engineers due to it being the best course of action for the environment, in addition to being a new economically viable alternative. Most suitable replacements for dipolar aprotic solvents or alternative synthetic strategies that avoid their use are often not used due to legislative and corporative pressures. These solvents are most often used in aqueous workups and produce mixed aqueous and organic waste, which can be difficult and expensive to separate. The wastewater that results in this requires extensive treatment and has an extremely high chemical and biological oxygen demand and requires incineration, which produces pollutants. Due to the ridiculously high amount of solvent used per kilogram of API, around 80 to 90%, even small reductions or improved efficiency in terms of amount of solvent used have large effects. For example, a 0.25 kilo reduction in THF equates to about a 4 kilo reduction in CO2 equivalent greenhouse gas emissions. This is before accounting for the disposal or recovery related emissions. In conjunction with reduced organic solvent load, membrane processes are especially effective in treating pharmaceutical wastewater and can be adjusted based on particle size of the contaminant. While membrane processes are extremely energy efficient compared to more commonplace separation and treatment techniques such as incineration, the issue that arises is that startup costs can be expensive and processes generally lack appropriate design to account for factors like pressure drop, concentration, or temperature. Altering the chemical level of drug manufacturing is another process modification and material substitution option for pharmaceutical companies. Here we have a graph showing two mechanistic approaches for producing celecoxib. The bottom is the launch process used by a manufacturer and the top is the improved process. The product yield has increased and the waste has decreased. Reactor efficiency has also improved, and product isolation requires cooling to 20 degrees Celsius, thus improving energy usage efficiency. The improved method resulted in reduction of organic solvent use, reducing 5,200 metric tons of solvent waste annually. This does not account for the reduced emissions that would be used for treatment. Highly concentrated waste streams were treated via distillation before being sold to a third party as generic solvent.
There are a variety of alternative waste treatment methodologies for the pharmaceutical industry. Biological treatments use microorganisms to break down waste mixtures into sludge, using methods such as constructed wetlands, vermicomposting, aerobic and anaerobic digestion, bioaugmentation, biotransformation, membrane separation, and irradiation treatment. These biological treatments require strict environmental control and monitoring, which include pH levels, temperature, rotational velocity, oxygen levels, and substrate concentration. Otherwise, there is a high risk of toxicity occurring. The most appropriate methodology for pharmaceutical waste treatment is membrane biological reactors. This is because they are effective at removal rate of pharmaceutically active compounds in wastewater compared to many other options. They are also eco-friendly, economical considering the initial installation costs, and easy to install and operate. The design of membrane biological reactors need to consider the following: plant footprint, effluent quality, process stability, energy consumption, as well as key design features regarding wastewater composition, volume, and temperature. There are 5 essential design characteristics that need to be met: pre-treatment, membrane location, membrane configuration, and systems operation, as well as downstream treatment. For membrane configuration, material type. Ceramic is the best but is the most expensive. There needs to be consideration for the mixture of hydrophobic and hydrophilic materials. The surface charge needs to be a negative, and the surface roughness and pore size need to be considered. For pore size, porous, seeding, straining, and size exclusion regarding micro, ultra, and nanofiltration are built. There's also non-porous, solubility, and diffusivity options. For systems operations, it can be pumping or pressurized vacuum or air scour techniques. The final effluent disposal options for pharmaceutical industries include landfill, open burning, and incineration. Landfill is harmful if any leaching escapes the system, and open burning and incineration produce a lot of air pollution but also have a chance for spreading pathogens. The best future option is zero discharge. This report recommends a few options to be taken for further studies in the future. We need to know the effects of long-term life cycle for unaccounted pharmaceutically active compounds and excipients that enter aquatic and terrestrial environments, as well as improve the toxicology testing methods. We also need to encourage policy reform to incentivize green chemistry and invest in better final effluent options.